Okay, moving on to section 5, we are going to talk about our final formed element, which is going to be our platelets. Again, this is also not a complete cell, just like our erythrocytes, because it's just a fragment or a tiny piece off of a much larger cell called a megakaryocyte. Even though it's not a complete cell and it doesn't have a nucleus and your typical organelles, it does contain granules that have a lot of chemicals in them. So some of these chemicals include serotonin, calcium, enzymes ADP, and PDGF, or platelet-derived growth factor. And these chemicals are really important in the clotting process, as you can imagine, because that's one of the functions of platelets, is to form a platelet plug and cause coagulation. So we have platelets circulating in our bloodstream constantly. We don't want them to start forming a platelet plug or inducing blood clotting unless we have some sort of blood vessel tear. So while they're circulating, how they are prevented from sticking together is by two chemicals. One is nitric oxide and the other is prostacyclin. So if we have healthy endothelial tissue or cells lining our blood vessels, meaning there's no damage, these healthy cells will secrete nitric oxide and prostacyclin, and those two chemicals prevent them from aggregating or sticking together. So let's talk about their formation. So their formation is going to be regulated by a hormone. That hormone is thrombopoietin. So when we talk about erythrocyte formation, that was regulated by a hormone as well. That was erythropoietin. When we talked about our leukocyte formation, that was regulated by two chemical messengers. One was colony stimulating factors and the other one was interleukins. And now we're on platelets and it's regulated by a hormone, and that's thrombopoietin. So we're still arriving from a hemocytoblast or hematopoietic stem cell, same thing. We're going to go into a myeloid stem cell, and from there go on to a megakaryoblast, which eventually will become a megakaryocyte. So there's not specific cell names for this developmental pathway. They just call or label the megakaryocyte in increasing stage number based on how large the megakaryocyte is. And I'll show you that on a slide on the next page. They don't really have a long lifespan, so they age pretty quickly and degenerate after about 10 days. So here's the pathway here. Again, you can see we're starting with the same cell that we started with all of our formed elements, and then we're going to become a megakaryoblast, and then a megakaryocyte, stage 2, stage 3, megakaryocyte, stage 4. That's about as large as we get. So that's the largest type of megakaryocyte we have, at which point it's going to form these cytoplasmic projections that are going to protrude into the lumen of capillaries. And when they go into the lumen of capillaries, the little extensions or projections are going to start to break off. And the pieces that break off are going to be our platelets. So that's what you can see over here. You can see that there are like little purple dots inside of the platelets, and that's going to represent the enzymes and the chemicals that they contain that are important in the inflammation and clotting process. So a good overview slide of all the formed elements now that we've covered. Again, you don't need to know specific numbers, not important. You do need to know the relative abundance of our leukocytes, and you don't need to know their lifespan really either. Just kind of understand what the function is, a little bit about the description, physically what the cell looks like. That is primarily for lab though. However, I would go ahead and learn it for lecture just in case.